technology. We just released this product. It's a VariantPro capture technology. And as we all know, in recent years, this target capturing technology has become a significant tool in focus genomic studies. But if you look at all the available methods out there, you know usually the target capture would require multiple steps. As we start to move this target sequencing method, or assay, move from research lab into clinical labs, there is a clear need to have a minimal hands-on step, have a simple process. So we developed this product or technology with this need in mind. And so in our technology, basically, it is one step of process. In this single step, we can start from, let's say this is genomic DNA, and you have target region you want to capture, and basically you just need to make your DNA sample with the kit we have, and in one PCR tube and one hands-on step. And after you put the tube on the PCR machine, after the completion of the reaction, you got your amplicon library prepared. So in this library, we put all the significant features in it, and obviously you would need a library primer that would be designed for the specific sequencing platform you want to use, either it is Illumina the sequencer or Iron type of sequencer. It can be just a matter of design those common primer or library primers. And obviously, we have these sample barcode or indexes there on both sides of the amplicon. And another significant feature we added in is a molecular tag. So what it is it would allow you to tag individual molecules in the starting samples, and at the end, when you get a sequencing result, you will get these multiple reads from the individual target sample and or target template, and then you can do a variety of applications on that. Later I will describe some details. And so here's what I'm going to talk about. First, I'm going to describe what the VariantPro capture technology is. And then I will show you some experimental demonstration data. And I do want to, since this is the beginning for us to launch this product, I want to spend some time to describe the design principle. So what we're hoping for is for users to have better understanding about the principle of this technology so that you can better utilize and take advantage of it. And so let's look at what the VariantPro capture technology is. So basically, this technology, we put in several innovations in it. And the first one is we call the Relay PCR. And what it is it's a fairly elegant PCR method. Basically, in this one PCR reaction run, and it would combine multiplexing PCR with it is multiplexing PCR. Basically, we combine the target capture with the multiplex amplification. So that we call Relay PCR. Later, with that term will become more clear why we call that. And another one is we have a very new primer design configuration. We call it Omega Primer. Basically, this is a multiple segmented primer, and it would give you a significant advantage of enhanced specificity and variant coverage. So we will describe about that later with great detail. And another one is molecular tag. It turns out the combination of our Relay PCR and Omega Primer, it becomes a perfect vehicle to put that molecular tag into your amplicon. So I will go through these things individually. And first, let's talk about Relay PCR. For this Relay PCR, in one PCR tube, we put these specific primers and the common primer and the target sample for the genomic DNA into one tube. So the relationship between these primers is like this. So this is a specific primer, and it has this specific section. So in complementary with your template DNA. So and then it has a tail. This tail is kind of like a common primer section, and this tail is the sequence is identical as part of this common primer. In the common primer, you have this index for multiple sample pooling, and obviously you have this tail section that can be made tailored to either you have different, either you have a Illumina platform or some other, or iron or some any other kind of sequencing platform. So this is a common primer. One of the significant features in this process, in this mix, is that we make this common primer, that concentration is significantly higher than this specific primer. And so how high? It's orders of magnitude higher. And it will become clear why we need to do that. So in this process, basically at the beginning, you have these specific primers, and they will be complementary to your corresponding template, and we'll do polymerase extension. And then after 2 cycles, you put this common primer flat at the end of your, I would call it PCRC. So basically here, in 2 cycles, you will complete capture process. After that, the common primer, before that, you do not have any complementary sequence in your template, because the common primer is really designed would have no homology with the sample you are going to use. If you use human, you would design this section to have the lowest possible homology. And so therefore, this common primer will not do anything to your anything significant to your genomic DNA until you incorporate this common primer in it. Then you have this common primer flat on both sides. And after that, then this common primer will take over the amplification. And why it would take over? Because the common primer, as I said, the common primer concentration is orders of magnitude higher than your specific primer. Just by simple computation, you would know this common primer will dominate the amplification process. Since it's order of magnitude, it's essentially it's just in the product, you mainly have the product flanked by the common primer. So that's how it works. And this process is really automatic. It's like a relay. The first section we call phase 1 is specific capturing, and then it's a monoplex. Now automatically switch to monoplex amplification. So that's how this works in the relay process. Now talk about the Omega Primer. This Omega Primer, it has 3 sections. The first part is the 5' on the 5' end, it has this 5p arm. And obviously, this 5p arm is complementary to your template. And then the 3p arm is also complementary to your template. And the difference is the 5p arm, it acts as anchor. So basically, it searches for the region designed for and stay there. And then the 3' we check if it is really correct. The nearest neighborhood is it correct, and separate by this loop. And if you want to, the second start from for amplification, if you want to use common primer, obviously this loop you can put the common primer section in it. And And obviously, we can put a variety of features in it for a variety of functions. Later, I will talk about more, what else we can put in. So it gives us really a flexible way of tailoring the function and the property of this primer. And the specific advantage of this type of primer is the first one, I would say specificity. So obviously, you compare, we all know the hybridization probe, let's say the hybridization capturing, and the advantage is that it's quite very tolerant, and the disadvantage is that it may not be that specific. So so when we put this thing in it, we have basically it's like by itself, it's like PCR, primer PCR, in PCR process, you have to have a forward primer, reverse primer, both based on hybridization principle. And you have to have both sections correct in order to get this primer functioning. So with two sections, obviously you can improve the specificity. And then the ability to tolerate certain template variation, that is because we make 5' arm long so that it can tolerate some variations. Later on, I will describe how we do it. It's the design principle there. And then another thing is reduce primer formation. And most people who have done the multiplexing PCR will know the dimer formation is one of the significant issue in the multiplex PCR. And here, the most significant primer crossing will be the 3' arm part. And this part is that it's short. So since it's short, it has less chance for the 3' arm to cross the 3' arm. And then another thing is the native sequence in the amplicon. So when you do the amplicon amplification, and after that you want to do the sequencing, obviously you want to have your primer as short as possible in your amplicon. Otherwise, in your amplicon, if it's a relatively short amplicon and you put a lot of section is due to your primer, then your useful length in your amplicon will be reduced. So obviously that's not what you want. And in this case, the part incorporated into the amplicon only 3 arm, which is short. So therefore, you increase your available native sequence in your amplicon. And then here's how we put the Relay PCR together with this Omega Primer. And here again, the Omega Primer, it's this section, the loop section. Basically, in here I just draw it like this section is largely identical to the common primer 3' section. And here I did not put the sample index. Obviously, we can put sample index. Usually we do put sample index there, so they can put your sample together. And this is the tail section, common primer. And the process is the same. And we use a significant larger, higher concentration in this case than the specific Omega Primer. And that is really PCR, just as I described earlier. It goes through 2 phases: specific section capture and monoplex PCR amplification. So that's really combined advantage of both Relay PCR and this Omega Primer. It's a perfect combination. And obviously, at the beginning, we want to test out if in this type of design would that work. Obviously, here it demonstrates that, yeah, indeed, it works. So when we start to finish with computation calculation, design such a structure and then test out, that was really thrilled to us. The people in the lab, with such a design, it works, and look how clean it is. It works really well, very, very fast. And here it shows the comparison of workflow. Obviously, everyone knows there are quite a few methods out there, and here I just show the 3 most commonly used methods, and And what I want to compare is the workflow. And obviously, as I described for our Relay PCR multiplexing processes, it's just one hands-on step. And as a comparison, here I show the extension ligation PCR. So what it is it has this oligo probe. It hybridize to your template, and then you need to remove the loose oligos and wash out, clean it up, and before you do the extension ligation, to avoid those primer concentrations are fairly high, and so that you can, before you do this reaction, you want to remove it so that you do not have a primer crossing. That would be really a very messy cup of soup. And after that, you will, of course, you need to do some cleanup process and then do the multiplexing PCR. And so that's how it works. It would require 4 steps. And the second one I want to show is multiplexing PCR using a modified primer. And here, the first step is multiplexing PCR using this modified primer. And so that also after the multiplexing PCR, you would use enzyme or some other chemicals to partially digest the primer section to remove it from the amplicon you already produced, and do some end polishing or end preparation, and then you would light it adapter. So that adapter basically is just suitable for your sequencing platform of your choice, and put the library primer there to light it to here. This process requires 3 steps. And the next one I want to show is it's more like a regular type of multiplexing PCR. So basically, the first step is multiplexing PCR, and with the common tails, the free prime end of the primer, and then you do several cycles of that, and then you do the pool amplification. And still, it requires 2 steps. And obviously, for you to move your assay from a research lab, for these processes in the research lab probably is fine. And I have to say, the performance, each one of these platforms, the performance, if you look at the data out there, I would say it's quite adequate. But if you move from the research lab into clinical lab environment, and you know what you really want to reduce that hands-on step, minimize it to reduce the chance for error, and reduce the chance for contamination. So one step is much better than multiple steps. And if you look at the time you are going to spend on it, here 4 steps, and it's 1.2 hours. And here 3 steps, about 1 hour. Here 2 steps, it says 5 minutes. And of course, depending on how many samples you are going to use. Here I put 5 minutes because, to be honest, people work in the lab, and let's say if you only do 6 samples, and you want to take the sample out of the refrigerator, you want to thaw it, and then you want to mix things together, you want to do a 6 sample, to be honest, probably it's half an hour, but each sample probably 5 minutes. But if you want to do a whole plate, that would be in seconds. So I would say, 5 minutes, I believe it, but here I would say, 5 minutes, or more, if you have more samples, definitely seconds. That's how I would calculate. And after that, here I just talk about the reaction section, and after, of course, for each platform, for all the platforms, it will be the same, because you need to do the cleanup, you need to do the library, so especially when you want to do the multiple sample pool. So you want to do the normalization, and then you want to put all the samples together, and then it's a PCR ready. That's the library PCR ready. And then let me just show you some experimental demonstration data. And this is our early demonstration, proof concept demonstration data. And in this case, we designed 6 amplicons using Omega Primer. And so they have slightly different lengths for the PCR product. And then here we show the individual ones. This basically is it's a multiplexing PCR, just show the individual one, and indeed all the lengths, they are the relative position, they are all correct for this expected length. And then here we put them together. Obviously you would expect this back a little wider, and so clearly it works well. And here I show the data. It shows, for this one, we put 63 amplicon as one of our demonstration panel, and it's a select from cancer genes. And in this case, we have 63. If you look at what I plot here is, here you have multiple amplicons, and here we have the read, or the read number, or the count, the sequence count. And this one we put on the library, was put on the Illumina plotted form. And here you see the uniformity. Yeah, there are some variations, but if you calculate the uniformity of this, it's really pretty good, because the usual standard we use for calculation of uniformity is like this. We first will calculate the average, the count for all the amplicons you have here, and then what you want to count is you want to see how many of these amplicons would have a read count above 20% of your mean value. And if we use that calculation for this data, and the uniformity is 97%. And so here basically just this one, it probably dropped just a little bit below 20%, but still if we use this rule strictly, this is 97%. That's how people calculate. And then one thing I want to mention here is that here basically we designed this primer, we put them together without any justification or adjustment to the concentration. So basically all the concentration we put, the specific primer we put in there, they are the same. And for multiplexing PCR, most people would do multiple round iterative process. The first you want to do the multiplexing PCR, you want to measure the individual product, and then you adjust your primer concentration, and do the second round, then do another round, and until you adjust to make it uniform. Here, it's basically what we expect is we do the design, and then we put them together to the, let's say, a customer panel, we expect it to work. That's how it works here. So it's really direct from computation calculation to the assay. And so here, just put it together, summarize the type of product we are providing. So this is the technology, Relay PCR, and for capture technology, and number of hands-on step 1, handling time, hands-on handling time 5 minutes, and the reaction time 4 to 6 hours. That's just for the PCR, capture PCR, and multiplexing the number of amplicons we can do. We do the computation projection. So we projected we can do 20,000. So obviously, it will take a lot of oligos. And in the lab, so far we tested, like a couple hundred, several hundred in that range. We have tested, it worked well. And but potentially it can go to very high number. And then DNA sample source, we tested the frozen tissue sample. Obviously, that's best quality sample, it works. And whole blood sample, it works. And FFPE sample, it works well. And obviously, FFPE, the quality is ranked widely, and so that's the type of FFPE we work. It worked pretty well. And the DNA sample input 10 nanogram, so it's a very low input requirement. The number of tubes, if you do the, let's say, the amplicon size in the range of 180 to 100 in that range, we can, depends, if all the capture site, the various sites of interest, if they are separated like SNPs, and then the one tube is sufficient. And but if you want to do the tiling, want to do a large section, you want to capture a large section gene, and want to do the tiling, we will require 2 tubes. And but sometimes some people say, okay, I have really low quality FFT, I really want to have a short amplicon. In that case, then probably would need a little bit more tubes, in and probably up to 4 tubes. That will be sufficient to capture all the target section you want to do. And for the dropout rate for all the panels we tested so far, it's really 100% success so far. And so dropout rate we have observed it's 0. And the coverage uniformity it's 97%, as I showed you earlier. And for the next part of the talk, we want to talk about the design principles. And as a scientist, we really like to understand how this thing works. And the primer design method I want to mention here is I want to show you the difference between the calculation method, the basic difference between this calculation method used in our design and commonly used for regular PCR or regular or any type of PCR primer design. It's really with a basic difference. That's what I want to show you. I want to show you by example. And so basically, we know one of the significant aspects of PCR primer design is to make sure that we have a sufficient primer template binding. So usually we would call it annealing efficiency. You want to have your primer have annealing efficiency as high as possible. And then the annealing efficiency, what it is what's the fraction of your template is hybridized by your primer, the annealing temperature you are going to use. That is the annealing efficiency. So in our case, so we have developed a primer and amplicon design tools based on rigorous chemical equilibrium equations and thermodynamic calculations. And these type of calculations would allow us to quantitatively predict the annealing efficiency for individual primers. But as comparison, the currently used PCR primer designs, we all know when we design primer, the first thing we want to know is the first thing we want to decide is what is your TM, right? We always say, okay, my primer has a TM of 65C or 70C or whatever. It depends on the annealing temperature in one of the fields. You see the TM, and that TM method is empirical because it does not tell you by a TM alone. TM basically is a melting point. A TM alone, you do not know at your annealing condition what the annealing efficiency rate is. So you do not know exactly what's how much of your template will be hybridized by your primer. So you only know, yeah, it will be a high number, but you do not know what that is. So here I want to show you, the simplified version of our calculation. Yeah, here I want to show you some equations, but I want to show you this is a fairly simple one, and especially if you're a chemist. So basically, if you're a chemist, you would look at this hybridization reaction as a simple, the so-called second order reaction. So by second order, what that is here in this chemical reaction, and on the left side, you have your reactant. One of the reactant is your primer, another one is your template. And then in this hybridization reaction, you would form equilibrium, and then you form this primer template complex. Here under equilibrium condition, you have the reaction constant, Ka. That can be simplified by this equation. And then what you want to know usually is, so when you have your template and the primer, how much your template is converted into your double-stranded primer template complex. That's what you want to know. What's the percentage? So that percentage, we would call it, strictly, we call it association fraction. But from biologist or biochemist point of view, we just call it a needing efficiency. So that's the same thing as a needing efficiency. And then, so the needing efficiency is unknown. We want to set up some equation to solve, to find out what it is. And it's fairly simple. So with this equation, we have the so-called mass balance. So basically, the amount of mass you put in should be equal to the mass of your product. So this should be, added together should stay constant. So that's the so-called mass balance. And this here is our Fa. So that's a needing efficiency. Here is how much the starting template you put in. This at the end the product. This is the starting template, the primer concentration you put in. So these two numbers you know. And the Fa is what you want to find out. And then another equation here, a simple equation is the so-called reaction equilibrium equation. And so here you have your equilibrium constant. So the equilibrium constant is your product divided your reactant. And then you introduce all these terms to the left side. Here you establish your relationship between Ka and Fa. And these two concentrations you already know. And then how to calculate Ka, that one is done by thermodynamic calculation. What it is here in this equilibrium constant, it can be calculated in a very, very simple way using this formula. Here, this is e to the delta g. Here, the minus delta t. This delta t is the free energy of your primer template binding. And the denominator, you have the gas constant, you have your temperature. That is usually you would put your annealing temperature there. That number you know. And then now the only thing is how to calculate delta t. It turns out during time between the May 1980 to early 2000, there had been a significant amount of work by physical chemists in the field of calculating for if you have these two different DNA sequences, and then you want to calculate what's the free energy and free energy. A large body of work already done before early 2000s. And nowadays, we can just put those thermodynamic parameters in it and put our sequence in it, and then you can derive this delta G. And with variety configuration for this method, you can calculate the simplest case with these two sequences. You can calculate folding. You can have your perfect match one. You can have a mismatch one. Variety of the situations you can calculate. But of course, putting everything together is quite comprehensive. But here, I just want to show you the principle. And once you know this K, then the things become simple because in this equation, everything is known except this Fa. Only one unknown. It's a simple equation. You can solve it. Or in a case when you have your template concentration significantly less than your primer concentration, this equation becomes something very, very simple. So left side you have the Fa, right side you have everything known to you, and then you can calculate this Ka with temperature built into it. So with that, you can calculate your annealing efficiency at the exact annealing temperature you're going to use. So everything becomes quantitative. And then that's what the TM method. So for TM, usually what's the definition of TM? Basically, the definition of TM is after this TM condition, you have your template 50% covered or hybridized by your primer. 50%, that's the definition of TM. And so basically, that obviously there is a simple correlation. We can derive a correlation between the FA or annealing efficiency, which I described earlier, and the TM calculation. You can go to the website, you can correct the website, it would allow you to calculate TM, and you put your sequence in, you calculate your TM. But what I want to tell you is that the TM and your annealing efficiency, they are not necessary to be always one-to-one relationship. Here, I calculated 4 different sequences on 4 different regions of genome. And here, number 4, 10. 10 different sequences. And from here, immediately you can see that if you have different sequence, the relationship between annealing time between your TM is fixed. Then they come out with different annealing efficiency and annealing temperature here we are using. And here the way I calculate here is back up a little bit. The way is I'll assume let's say in PCR reaction I use tag polymerase. Everyone knows the tag, so use tag. And if you go to the enzyme manufacturer, and then they will tell you if you use our enzyme, the first thing you want to design a primer. You first decide what annealing temperature you are going to use. Then you use that annealing temperature plus 5. That is your TM. And then in this case, I say, okay, my annealing temperature is 65. That's why I set the TM to 70. And all these TM, they are fairly, here it's a TM from, in this case, my calculation, from 69.5 to 70.3. It's in the very, very range. And then if you look at the actual annealing efficiency at your annealing temperature, you see that FA ranges from the lowest one is 0.891. The highest one is 1. And then for multiplexing PCR, you will say, okay, here 0.891 is close to 0.9, close to 90%. It's not too bad. And in reality, probably with that primer, you can actually make your PCR work. That will work. And that's multiplexing PCR. But the thing is, if you want to put all these 10 primers into one tube and do your multiplexing PCR, and after that, you have 20 cycles. Let's say after 20 cycles, this one, then the normalized the yield is 1 because the efficiency, the binding efficiency is 1. So that's 1. And for this guy, so if you have 0.891, then after 20 cycles, what you have? You have your normalized yield 0.099. So the difference between this guy and that guy after 20 cycles, your amplicon yield difference tenfold. So that is one of the major reasons why in multiplexing PCR often we see the product concentration range really very, very wide. And sometimes it's a tenfold, sometimes it's not surprising, sometimes it's 100-fold. It's quite common. And so the design, how you design the primer matters. Indeed, matters a lot. And now we know how we can assess this PCR quantitatively. Now I want to compare the conventional multiplexing PCR with the Relay multiplexing PCR from quantitative point of view. So as most people know, in multiplexing PCR, there are two major issues. One is amplicon yield variation. Another one is primer dimer formation. These are two major issues. Let's break down these issues and look at what's the component cause these issues. Then let's look at the amplicon yield variation. What's determined by. The first thing I put here is the primer initiation efficiency. Basically, this is somewhat it's largely related to the primer annealing efficiency. Why it's not always the same? Because you can calculate your primer. You say you have the FA is 95%, let's say, and then your primer maybe the 3' end. What's the sequence you have there? That also matters. Maybe you have TC, then your extension can start pretty easily if you have a lot of AT in there. Even so, your annealing efficiency is close to 1. But if you have a lot of AT in there, it may not. Some of the primer may not extend. So that's why I would say the initial efficiency is less or equal to FA. And so that primer is one significant factor. And another one is the primer extension efficiency. What it is how much of your primer extended make the amplicon. So in between the primer, you may have some, in the amplicon, you may have some region, difficult region, usually we call it. And that region will be, let's say you have high AT. And then before your amplicon reaches to the other end, and then that like chain, it drops. So then it may not reach to the end. And another scenario would be you may have some secondary structure in the middle of your amplicon, especially when you have high TC content amplicon in there. So those are the issues. So therefore, the two main determining factors will be the primer and your amplicon composition. And so your amplicon efficiency will be determined by these two. And after the number of PCR cycles, let's say the PCR cycles are n cycles, and then you have the initiation, you do the n times, and the extension obviously do n time, and then here it would be exponentially dependent on your individual efficiency. That's your amplicon yield. And then another issue is the primer dimer formation. The primer dimer formation is determined by, of course, obviously this is when you have a lot of primers, it appears to be very complex. But if you break down into individual reactions, then again, this is simple. It's just the simple, the second order reaction I showed earlier. So in this case, the two reactions will be two primers. They could be the same, it could be different primers. Two different primers. That depends on the sequence design. Some of the primers may have longer overlapping than others. So that overlapping at the end, it will turn into K. So basically, you have two overlapping regions, you calculate your thermodynamic free energy, and then you can calculate K. That's the K. And then you have your concentration is second order reaction, then it's K times the C primer, C primer, and that's the concentration. So again, this is a breakdown into a simple second order reaction. Then you have multiple different combinations. You add everything together. That is your combined primer process. So if you break down everything, then things become very, very simple. And that is the beauty of actually, we are in the field of biology. If you look at the field of physics, that's how the physics solve their problem. They break down everything into very, very simple one. And nowadays, I see the biology, we are starting to go into that direction. Eventually, everything we can break down into simple components, we can calculate a lot of things. That's how we start to do it. And with this thing, we know, now we know we need to minimize overlapping between the primers. Another thing, if you can drop the primer concentration, and then your primer product amount will be reduced very, very rapidly. And that's how this thing works. Reduce the primer concentration, and as I said, in our Relay PCR, and two components here. One is here, you have initiation n times. In our case, specific primer, just one time. Each primer, you just do one time extension. So that n drop, that has a lot. Basically, you start from n, and in Relay PCR, this n becomes n equal to 1. And obviously, for the extension, then in our quantitative amplicon design, we will be able to quantitatively calculate the probability in between the primer. So also, we can optimize that extension efficiency. So, but that n stays, because you have to amplify n times. That you cannot isolate. You just have to do the best job you can to minimize that difficult structure in between. And so that obviously with this, the primer alone, we can improve the uniformity significantly for in the multiplexing PCR. And another thing, the primer-dimer formation, as I said, the primer concentration will reduce from the regular one by orders of magnitude. For each one, let's say here, you reduce by 1,000 times. To put together, you would expect reduce primer-dimer formation by 1,000,000 times. That's significant. So put together, indeed, we have significant advantage using the Relay PCR than the conventional multiplexing PCR. Now we show in a quantitative way. And now let's look at the specific primer. The specific primer, how this Omega Primer can enhance the specificity. And here, if you look at this three segmented structure, what it is I would use an analogy that this loop here is like a spring. Basically, what it is here, you have something, 5' arm have something hold to your template, and 3' arm, you hold it to template so that this spring will not pop. So that's how, and generally, by calculation, how it works. So that is, if you really look at the principle, it's a signal. You just want to balance the force, because if in this hybridization, it does not like this loop, so you just have to hold it there. And then, since here we use the minimum length of this 3P arm, if you have a little bit of defect in this 3P arm region, what happens is you would weaken the source to hold this spring there, and that spring would pop. And now if it pops, obviously you will not be able to have your extension to receive from 3' end. And that 3' end is not available for your extension reaction anymore. So that's why, just by looking at this structure alone, you would understand why it's more specific. And then people always, some people, they initially look at this, they say, do you have a rough number to tell me how many times it is improved? You say it's improved. Do you have any number quantitatively to tell us? Yeah, then we give a thought, then here's how we calculate it. So, our design, usually 3P arm section, the average length is about 16 nucleotide long. And then, let's say in a totally, we design the primer for totally random sequence. It doesn't matter what that sequence is, just totally random. Then 16 nucleotide, we have the probability of 4 to the 16 many sequences. And then, let's say we assume we can tolerate, the 3' arm can tolerate up to 4 nucleotides. Actually, 4 out of 16 is a lot. We put a lot of safety factor is there, but just for the argument's sake, we say we can, at least allow 4 mismatch in there. And then you have 4. Obviously here, you have 4 mismatch, you have 3 mismatch, you have 2 mismatch, you have 1 mismatch. You all allow. That's all allowed condition. And then your probability of forming a significant but a wrong binding will be calculated by this formula. That probability is 3.8 times 10 to the minus 5. And then if you ask me how many times, then I would say, okay, 1 over p, that's 26,000 times. That's how we do the data estimate. And that is statistically, that how many times we can enhance it. So, and then, so I just show you this Omega Primer, how specific it is. Then the next thing I want to tell you is that at the same time, it can tolerate a local variant. So why don't we need to tolerate the local variant for PCR primer? We always put the PCR primer into a region that has no variant. That's usually how we do it. We want to avoid it. Why do you want to do that? What I want to tell you is, yes, indeed there is a need. If you look at the whole human genome, and if you just look at the SNP database, and you look at the high allele frequency, above 1% allele frequency, you would be able to find there are 3 million of those SNPs in it, and 3 million the 6 million in it, and the 6 million out of 3 million of human genome, you have 1 SNP for every on average for every 50 nucleotide or 50 BP long. So that density is very high. And if we want to design a capture, and if we want to do the tagging, sometimes it's just inevitable. You have to run into that region. Or if you want to avoid it, maybe it's possible done by some other factors, and that region may not be bad. So that's why indeed there is need to be able to tolerate that variation. And in fact, some people, when they design panel, sometimes even a small panel. Let's say some people want to design one BRCA2 panel, and the whole all the exons put together probably around 200 amplicons will be enough to cover that region. 200 amplicons is a very small panel. And but some scientists, when they look at a different technology, they say, oh, but I do want to avoid allele dropout. So that's an important factor to me. And then when they look at it, they say, okay, PCR, I know it's highly specific, but I want to avoid allele dropout. Then they say, okay, maybe in this case, hybridization method using hybridization probe is better because it's long and it can tolerate variant. So that's the criterion they use. And then they would say, okay, they would argue, hybridization method is better in this case. But in our design, we have this consideration of variant tolerance built into our design. And here I show how we do the design. So basically, if you look at the reference SNP database, and basically you have, as I said, you have your SNP density very high. And that's actually, if you really think about it, in reality, that cannot happen in a single person. When you do your PCR, when you do the sample, you do one person, you do not want to, usually you do not want to do a mixed person, now you do not always own. And you want to do the barcode to do that. For individual capture, it's one person at a time. And for one person to put so many SNPs in there, I'm not sure that person will be viable. It's not real. So we want to, make something more realistic, and that we looked up. So basically, the database we use will be 1,000 genome to look at the non-pathogenic genes, non-pathogenic variants. So we use 1,000 genome. In it, in 1,000 genome, you have 2,500 individuals, and the sequence you can get the haplotype, and now you know in the haplotype what's the SNP location look like. And then we obviously use the 1,000 genome for general population, and then combined with user-provided pathogenic variants. For both innate and preferably, you have also the individual from the individual person. Initially, all the database you can find, you can indeed find the individual record and then the individual in. And then the way we design it is we put all these individual, either it's an individual or haplotype, and then we have SNP position in our design. We look at all these things. We want to make sure our 5' can tolerate the worst case scenario of all these variants. When you can handle the worst case scenario, you can handle the real person individual. And the good thing is, in our design, the design is produced with a predictable population coverage. That's a significant. There is a statistic prediction in it. You can predict. It won't work. You can design it. You can, if there's a database, you can say that you want to do the 1% of a population, or you want to do, let's say, 0.5% of population. So if you can choose that, you can provide statistic prediction data. That's an advantage. So in this case, so what this is 5' is start to localize the hybridization probe, and that is really combine the advantage of hybridization method of variant coverage with the PCR advantage of high specificity. You can combine the best of both worlds in this design. So that's why we're so excited about this type of structure. And then I want to show you, how we do for this type of, Omega Primer, how we look at the primer crossing. And obviously the crossing is significant here. And the way we look at it in calculation, we actually build the method to look at different scenario here, just to show the typical site, the typical types. One is if you have a crossing between the 2, 3' arm section, and if one of them, at least one of them is expandable, the product of this will be the most harmful one, because in this product you have 2 common regions, and that product will be amplifiable by your common primer. So we will build some very, very stringent factor to look down this thing, to filter out that type of primer. The good thing is the 3' arm section is short, so the probability of these 2 overlapping is statistically low. So then another scenario will be if you have a 3' section overlapping, but the very 3' end of your primer is not binding. In that case, you will not have the extension reaction. The end of essentially not too much harm done there. And another thing I want to show you is, here in this case, 3' arm is extendable in the 5' arm region, but this product, so you may not like it too much, but what I want to tell you is, don't need to worry about it too much because this thing is not amplifiable. And your specific primer concentration is so low, the amplification efficiency there it's not that high. It can do good job for binding, for capture, but this type of thing, the amplification is very high. And the next thing I want to show you is how we put a molecular tag into this amplicon. The first thing I want to describe a little bit about the molecular tag, just in case not everyone is familiar with this. What this is regularly, for molecular tag, there have been a few publications to describe it. Regularly, what people do is people would, start with, the first step is fragment your DNA sample, and you fragment it, and then you use your adapter. You design a special type of adapter, make a 3' end of your adapter to contain some degenerate sequences. So once you have degenerate sequence, here is a comment, here is degenerate sequence. Once you have this degenerate sequence, at this end of your adapter, right after the ligation, once you put it in, you actually put a lot of degenerate code into your amplicon. And the first step, since you have so many different degenerate code innate, essentially when you look at the individual molecules, the chance is that your individual template molecule, each one will be tagged by a unique, as a pair or single one, a unique tag. So by that, after amplification, if you have read number high enough, for each amplicon you have multiple reads. Those multiple reads, if you look at these combine these unique tag together, you would be able to identify, trace back to your starting molecule, template molecule, one at a time. So that is really good feature for that. But the thing is, in the current available thing, the available Omega, here if you really think about it, how you can put this degenerated segment into your primer. It's doable. It's quite involved work to make such type of primer. It's quite involved. It's not so easy. And also here you need to do the fragmentation, you need to do the ligation, you need to do then you need to do PCR. Obviously, it's a multiple step. And the good thing is, here as I said, the sequencing method we are using now, just by using this standard high throughput sequencing, and then in the result, and your sequencer, it becomes a molecular counter. So you can count how many molecules in there. You can do a quantitative profiling. So that's one aspect of it. Another thing is you can correct error. This sequencing, high throughput sequencing method we look at it is even for the, for example, let's say aluminum platform, we all know it's pretty good. The error rate compared to some other platform is said to be pretty low. How low it is? It's about 0.5 to 1%. It's not too bad. And but if you look at your amplicon length, as I said, many people do 180 to 200, and if your probability of error rate of your sequencer is 0.5 to 1%, inevitably the chance is you will have at least one error in your rate. So then, but if you know, you can trace all back to the individual molecule, So I have these things, you look at it, and this one will be concordant, and these things are not concordant. Then you can do the analysis, you can filter out all these sequencing or PCR induced error out. And that will make your error rate, you can reduce your error rate significantly. And then the calling rate, the sensitivity will be significantly improved. And in our case, how we can introduce this tag? It's very simple because, as I said, in our case, the specific primer, in this case, Omega Primer, they are functioning only in the first two cycles. And then you can simply put this molecular tag, a degenerate sequence, into your Omega Primer. Then just one step, one hybrid step, one PCR reaction, you have your molecular tag incorporated into your amplicon. And then another thing we want to do is we put a lot of design into this amplicon, into this molecular tag. Molecular tag, most people they just put an in there. But then at the end, you look at the sequencing result, you would realize. Then I put, let's say I put 6N in there, then the 6N you would expect, you have 4,000 different ones. And then at the end you see the distribution of these 4,000 different tag sequences, they are not quite the same. They are sort of like there's a PCR bias in it because 6N, if you look at it, if you look at the folding all that, it's pretty long. And then you cause a lot of folding, therefore your PCR is selective, and then it would amplify those ends without folding better than some others. So that's why you have those bias, PCR bias. We put a lot of design. The way we do it is we put degenerated sequence in between we put non-degenerate sequence between the degenerate sequence. And with this design, we can make sure that we have minimum folding for these primers. And also we minimize the probability of crossing hybridization. So we put all that consideration in it. And then to summarize, so basically we have developed a novel multiplexing PCR in combination of Relay PCR and Omega Primer. And the characteristic of this method is one-step reaction and local variant tolerance, high specificity, and molecular type compatibility. One comment I want to make is, also here I show you, the capture for sequencing. One thing I want to say, this is a general applicable multiplexing PCR method. It can be used for a variety of applications. And so if some of the audience may have some other problem, you can think about it. It does not have to be the capture problem. It can be any assay involving multiplexing PCR. That concludes my presentation here. Thank you very much.